Okay, good morning everybody. We're going to start off today with our first lecture on this Unit 5, DNA and Cell Division, and today's lecture is going to be on the cell cycle, which is a type of asexual reproduction. So let me go ahead and get ready. Once again, as far as in the textbook, in the Big Campbell, check these chapters out because I think these go with my book, which is the newer edition, but we're dealing with 3 different chapters with this. If you're using the Hillis, it's chapters 7 and 9. All right, so let's go ahead and get started. This part of it is we're dealing with asexual reproduction. I'm going to see if I can get my Mobi up and running. Here we go. All right, so asexual reproduction, which is, you remember we talked about in the beginning of the year, asexual means no exchange of genetic information. Okay, or no exchange of DNA. So we're going to end up with identical offspring. Okay, no mixing of DNA. This happens not only in unicellular organisms, it also happens in multicellular organisms such as us. This is the primary way that we make more skin cells or more muscle cells if we need them, or more stomach cells for the stomach lining. So this is going to happen not just in unicellular organisms but also in multicellular organisms. But the end result is we will end up with 2 identical cells in terms of DNA content. Okay, so same number of chromosomes, same amount of DNA, everything should be identical between these 2 cells as a result of asexual reproduction. Okay, the purpose, if it was in unicellular organisms such as bacteria, which is our 2 kingdoms, archaebacteria and eubacteria, okay, or also for yeast, which are in kingdom fungi. So with archaebacteria, eubacteria, or kingdom fungi, and then we could also talk about our protists that are unicellular. So any unicellular organism, when they go through asexual reproduction, the whole big picture is they end up with new organisms. So the benefit for them is not to just make new cells, they actually get new organisms because of the fact that they are only unicellular organisms. So it's their primary way of reproduction. Okay, for us, not so much so. For multicellular organisms, which would be us, this would be plants. Okay, this is plants, animals, other multicellular organisms like protists. Okay, there are some multicellular protists. Organisms that are multicellular, when we go through asexual reproduction, the primary reason is for growth and development or renewal and repair. Okay, so for growth in terms of, like in the case of when you're growing and you're getting taller, you need to make new osteocytes, which are bone cells. In the developmental process to gain more cells, just let's say new skin cells, or to replace the stomach lining, or to replace any kind of cells that might be damaged. So for development, for growth, and then also for renewal and repair. All the time we are constantly replacing skin cells. Those are the ones that divide the most, just from normal wear and tear because we slough them off on the outside, but also for renewal purposes. So renewal, repair, growth, and development is the primary reason that multicellular organisms go through this process. But whether it's unicellular, multicellular, the whole purpose is we will end up with 2 cells as a result that are identical in terms of DNA content, amount of DNA, exactly what they look like. They share the same number of organelles, etc. All right, so for a second, before we get to us, we're going to talk about prokaryotic asexual reproduction, which is called binary fission, because we want to compare prokaryotes to us. I think it's important to look at one of the major differences between them. First of all, this is a very fast process, much faster than when you compare this to a eukaryotic organism. Think of some of the things that we've talked about, though, in the past. If they're prokaryotic, they lack a true nucleus, and they lack membrane-bound organelles. So that means there's no mitochondria, there's no lysosomes, no Golgi, no ER. They don't have any of those organelles that are membrane-bound. Remember, the only organelles they do have are ribosomes. So when you look at the time that's required for this type of reproduction, this binary fission, it's very, very quick. Okay, they also don't have to deal with a nuclear envelope. When you're talking about eukaryotic organisms, such as us, if we're going to be doubling the DNA and splitting the cell in 2, that means we are dealing with a boundary. Okay, we're dealing with that nuclear envelope boundary because that's where the DNA in us is located, is inside that nucleus. So for them, they don't have that problem of dealing with a nuclear envelope because they don't have one. Okay, you can also look at how much DNA they have. Their DNA, they simply have 1 circular chromosome Okay, they have one chromosome. It's in the circular form. You can see it inside this picture, in the first picture right here. It's this big, long strand that you see listed in here. Okay, that's one circular chromosome. It is double-stranded, just like ours, so it is a double helix. So if you're talking about comparing a prokaryotic chromosome to ours, instead of having circular chromosomes, we have linear chromosomes, and theirs is one circular one. We have 46 total. They have one, so the amount of DNA is much greater in us when you're talking about humans. So they don't have what we call the cell cycle. Their cell cycle is much shorter, and so we give it a name called binary fission. Okay, and you can see in the picture what's going to happen in binary fission. It's kind of a really quick process, but they have their single chromosome is going to replicate. Okay, so it doubles. Just like in us, they have to double the amount of DNA. If you want to end up with two cells at the end that have the exact same number of chromosomes or the same amount of DNA, they're going to have to double what they start with. So in this first picture, between, I would say, here and here, you've got doubling of the DNA. Okay, so it makes a copy. So you would have this, and you would have a double amount in here, 2 times the amount of DNA. Okay, each copy, once it's doubled, actually moves to the opposite ends of the cell. Okay, can you think of why that would happen? Hopefully you just said to yourself, so that at the end, each new bacteria is going to end up with one of those copies. So one of the things that they do is when they make the double chromosome, each one starts to move to an opposite end. Okay, there's also some research that shows that one of these, like this chromosome right here, has a slight attachment to the cell membrane right inside the cell wall to hold it to this side of the cell. This one does the same thing, has a slight attachment that holds it to this side. That ensures that when this thing splits in two, that each of the new cells is going to end up with the copy of each chromosome. So you can also see, like between here and here, that we've also got some elongation happening. Elongation means some growing. What are we growing? They have to grow some cytoplasm. Okay, cytoplasm, which means the cytosol, which is the cell gel, plus they also have to double, their one and only organelle is their ribosomes. Okay, so they're going to be doubling and elongating at the same time. Once the bacteria is 2 times its original size, then it starts to actually pinch inward. Okay, now remember they do have a cell wall, okay, but their cell wall is made up of mostly polysaccharide material, so they'll still start to pinch in right in here and form new cell wall material, and eventually they will split and you'll have 2 new bacteria. Okay, now technically we're going to call this cytokinesis, which is division of the cytoplasm. Okay, so even though when we compare this to us, they do not go through phases such as interphase, prophase, metaphase, anaphase, and telophase. They don't have any of those phases because theirs is called binary fission. However, cytokinesis is defined as division of the cytoplasm. They do have cytoplasm. So they are going to split the cell into 2 by cytokinesis, which is one commonality between prokaryotic and eukaryotic organisms. Okay, but by the time you're done, this is a whole new organism, okay, right here, and that's a whole new organism here. So because they're single cellular, they just reproduce to make whole new organisms. That's not how us, as eukaryotic organisms, we do not reproduce and make more like this. We use sexual reproduction to make more. Okay? All right, so that's binary fission. You can see the last bullet here is there's 2 identical cells produced, and like we said, whole new organisms. All right, little picture. I thought I would throw this in here for you. This is kind of cool when you look at how this is happening. You can see this is a true electron microscope picture. Here's the DNA in this one. There's the DNA in this one, and you can see the cell's already gone through cytokinesis. So these are rod-shaped bacteria. If you remember, that's bacillus. This is going way back to Unit 1. So there's a rod shape, whereas in this picture, this is coccus bacteria, okay, and you can see the DNA is right in here. And they can reproduce so quickly, I wanted to kind of show you that this is the first cell division is between this one and this one. So there's cytokinesis that's fully happening. But they can reproduce so quickly that this one is already reproducing again here, and this one is also reproducing here. So the speed at which they do this is very, very quick. Okay, in a matter of about 20 minutes, they can go from 1 to 2 cells. If you compare that to eukaryotic, as far as our cells, human, some of our cells take an entire 24-hour time frame to reproduce. Others take years to reproduce. So for bacterial binary fission, it's a much more rapid process. All right, well, let's get to us, because of course we are the most important things, so we have to talk about us for a little bit. So with the eukaryotic cell cycle, okay, we call it the cell cycle. The cell cycle is defined as all of the events that take place from the beginning, B-G-I-N-N-I-N-G, from the beginning of one cell division to the beginning of the next. Okay, so it's all the events that take place from the beginning of one cell division to the beginning of the next. Okay, I'm going to pause for a second because my dog is going crazy. Hang on. As you can hear, that's her. Okay, sorry about that. Okay, so anyway, going back to the description of the cell cycle, like I was saying, it's the beginning, it's all the events that take place from the beginning of one cell division to the beginning of the next. It can kind of be described as the life cycle of a cell, because it's when you start off and you have, oh, then here's the first cell, it's all the events that take place until that cell decides that it's going to divide into two new cells. So we call that the cycle of the cell. Okay, it's divided into two major parts. Okay, and this is what I was telling you guys about one of my big beefs of calling this. I do not like calling this chapter mitosis, because when you do that, you're limiting yourself to these PPMAT. When you simply call this chapter mitosis, or this part that we're talking about, then that means you're only talking about prophase, prometaphase, metaphase, anaphase, and telophase. And there's actually a big part over here at the beginning, and there's a big part down here at the end. So all of the phases in the entire cell cycle are interphase, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. And so you can't just call it this one part, although that one part, mitosis, is defined as division of the nucleus. Okay, make sure you write that down. So mitosis is defined as division of the nucleus. Yes, that's very important, and it's a huge part of the cycle. But C, as we've already talked about when we did binary fission, is cytokinesis, which is division of the cytoplasm. Okay, so that's very important. Dividing the nucleus is important, dividing the cytoplasm is important, but hello, look over here at the big fat I. It is very important to go through the three subphases, which are called G1, S, and G2. And we're going to explain these a little bit more on the next couple of slides. But if you leave that part out, you're leaving out the big S, which is copying DNA. So we can't leave that out, okay? So one of my biggest things when I teach this unit is I like to use the term the cell cycle. It's all the events that take place, including interphase, which is G1S, G2. Yes, definitely including mitosis, which is division of the nucleus, but we also have to include cytokinesis, which is division of the cytoplasm. So there's lots of steps that take place in the cell cycle, which is why this diagram is a really, really good diagram to use, because it puts everything in this one circle. Okay, so if you're just going to mark it off as like what to call everything, you can see right in here, interphase is listed as a big main part. Okay, and like I said, that's subdivided into G1, S, and G2, and we will talk more about that in just a minute. Okay, but then they put it into M. Look at M. M is called the mitotic phase, and yes, it includes PPMAT, but it also includes the C in with that. Okay, so the cell cycle alternates between interphase and the M phase, which is the mitotic phase, and yes, that also includes cytokinesis within that. So if you look at your diagram, so let's kind of break this up a little bit. From here, all the way around, okay, over to here, that entire thing is interphase. So I'm going to label it like that. Okay, within that, we go G1, S, G2, okay, like that in the cycle. Now that actually, guys, I know this may not be drawn to scale. I mean, I think they tried to, but that is 90% of the cell cycle. The cell spends in interphase, okay, one of the three subphases. Now G, just so you know, stands for gap or growth. It depends on what you're looking at, but it can stand for the first gap. Some textbooks call it like growth because the cell is doing some growing. So the G2 is called the gap 2. The S stands for synthesis. Reason being is synthesize means to make, What are we making? We're making DNA. So the cell is going to copy its DNA in S phase. So that's a very, very important phase as far as interphase. So G1, S, G2 are all part of interphase, which is in this big time frame. Okay. The smaller place in here, okay, this is when in this section you've got mitosis, okay, and then you've got cytokinesis. Okay, so inside the mitosis part, that's when you have PP-mat. I like to say PP-mat. Prophase, prometa, meta, ana, and telophase. And then, of course, cytokinesis. So we can put all that into the M phase. So if you look at the whole cycle all at once, it goes from interphase, G1S, G2, around to M phase, which includes mitosis, PP-mat, as well as cytokinesis. Okay? So you think you got the cycle? Okay, that's the big overview. All right, let me get rid of that. Okay, so let's go through these a little bit more in detail and talk about actually what's going on in each of the phases. Okay, so we're going to start off with interphase. Okay, a couple of these are just general bullet points before you get into the specifics. This is the part of the cell cycle in which the cell is carrying out normal activities. Okay, what do we mean by normal activities? Let's talk about some of the things that the cell would do. How about cell respiration? We just talked about that in the unit. That means making ATP. The cell's going to definitely need to make its own energy source, so that's a normal activity. What else is a normal activity? Protein synthesis. Okay, making proteins. Because we are definitely going to need a lot of proteins for cell division, but it's even going to do that just if it's not going to divide, it's still going to go through and make a lot of proteins. What else would be a normal activity? How about replacing organelles? Okay, maybe a mitochondria is not functioning anymore and we need to get rid of it, so the cell will take care of it, recycle it, and replace it by making a new one. So all of those activities are going to happen during interphase. Okay, like we said a minute ago, about 90% of the normal cell cycle is spent during this phase, because that's a lot of activity just to do those three things that I've listed there. Okay, just to do normal activities in the cell takes a lot of time. Okay, DNA is inside the nucleus, of course, but the form of it is called chromatin. Okay, chromatin. The best description for me is if you look at spaghetti, if it's laying in your bowl, it looks like this. It's very loosely arranged. It's just kind of like there. It's not really condensed into chromosomes. It's very loosely kind of laying around with its associated proteins. It's got a lot of, I'm going to put dots here, a lot of proteins that are around. So chromatin is defined as DNA plus protein. The proteins specifically are called histones that are very loosely arranged inside the nucleus. Okay, so we're going to refer to it as chromatin, especially during interphase. Now, if I talk about like the other words that describe DNA, okay, one of the words that I want you to know is called genome. Okay, genome is the total endowment. Okay, endowment of DNA. What do I mean by that? Each species has its own genome, its total endowment of how much DNA it has. For humans, our total endowment is we have 46 chromosomes in each of our cells. So in a diploid cell, okay, diploid cell, which means has a double set of chromosomes, we use the term 2N to describe that. For humans, 2N equals 46. Okay, that's a diploid cell. A diploid cell would be a somatic cell, okay, such as example would be skin cell, okay, a muscle cell, a nerve cell. It's basically every cell that we have, because somatic means a body cell. It's every cell in our body except for sex cells. Okay, sex cells, because they are supposed to come together to form a new organism. Sex cells are the cells in our body that are called haploid. Okay, so we use the term N to describe those. So in a human diploid cell, our total endowment of DNA is 46 chromosomes. Okay, in an organism such as a fruit fly, why did I choose that? Because fruit flies are very important for genetic studies, which is our next unit. Their total endowment is 8. So they don't have as much DNA compared to us. Okay, a monkey, okay, a chimpanzee, for example, their 2N number is 48. So each species has a different number that's unique to the species. That's the total endowment. So for humans, like I said, ours is 46. Okay, what does that mean? Well, if you look at where those came from originally, we all started off as one cell big. When the sperm from your dad met the egg from your mom, those came together, each carrying 23 chromosomes because those were sex cells that were haploid, and when they joined together, that created you, which you started off as one cell big that had 46 chromosomes. Okay, so inside your mother's womb, that one cell then started to go through the cell cycle to make more and more and more that all had 46 chromosomes. So that's how all of your cells ended up having the full set, was because we all started off with 23 from mom and dad. Okay, so that kind of makes sense. I know there's a lot of information there, but I wanted to describe the word genome is total endowment of DNA for each species. Okay, and then when you look at, let's say we use the word, I'm going to use a blank screen here. When you use the word chromosome, anytime you use that word, just so you know, chromo means color. Some means body. So the reason that they called chromosomes is because they can be stained with a certain color stain that makes them appear under the microscope. Okay, so when I use the word chromatin, that's still referring to DNA plus protein, but it's loosely arranged like this, meaning that it's indistinguishable under the microscope. You can't see it as a chromosome. Under the microscope, a chromosome is seen like this, if it's by itself, or if it's in its sister chromatid pair, it's seen attached together, like that, if it's in sister chromatids. Okay, so we're going to be using a lot of different terms that refer to the DNA, the genetic material inside the cell. All right, going back to this slide, there are 3 subphases of interphase. Okay, you can see here G1, S, and G2. Okay, so G1 is when a cell is not yet dividing. Okay, this is not yet, there's no decision, there's no cell signal for it to divide yet. G1 is just a normal growing type of activity phase where the cell's making proteins, it's making ATP, organelles are replaced if they're needed. Okay, the cell hasn't gotten a signal yet. Okay, if the cell receives a signal that says, hey buddy, I need you to divide, then the cell switches and clicks into S phase, which S stands for synthesis, okay, where DNA replication takes place. Now this takes a lot of time, y'all. You can imagine, like, out of a 24-hour time period, if it takes a cell 24 hours to divide, this synthesis part is taken upwards of like 10 to 12 hours. This is a very long process because all of your total endowment, your 46 chromosomes, must be copied during synthesis. Okay, it's kind of nicknamed also the point of no return. Okay, that point of no return is actually between G1 and S, because if the cell passes that, it's already made the decision that it is going to go forward and not return backward. It's going to go into S, it's going to go into G2, and then it's going to go into mitosis. So that's why it's nicknamed the quote-unquote point of no return. But like I said, that point of no return checkpoint is about right here. It's between G1 and S. So it takes a signal that tells the cell to do that. Once it clicks and goes into S, it's going to replicate the DNA. Okay, now we will talk about this process of replication in much more detail at the end of this unit, so that'll be a different lecture. Okay, but that's basically, just imagine that the total amount, the total endowment of chromosomes will be copied. Why? That's so that we have enough to fit 2 new cells. Okay, after S, it clicks into G2. Okay, G2 is a major preparatory phase. Okay, what is it preparing for? It's about to go into mitosis, where it will divide the nucleus. So if we're going to divide the nucleus, we have to prepare to do that. So we have to prepare how? Because we have to double all of the organelles. If the cell has, let's say, 50 mitochondria, it's got to double those 50 so that we have 100, and each new cell will end up with 50. So all the organelles, the mitochondria, lysosomes, Golgi, ER, smooth ER, rough ER, all those things are doubled. The centrioles, if you remember, we hit that way back in the unit we talked about the cells. The centrioles, we said we would talk about them later, those are doubled as well. Okay, the reason being is that's where the centrosomes are located, which are going to form the spindle fibers. So we're going to have to form spindle fibers, so that's going to need to be in each of the cells. Okay, so in this picture, this is what you guys are going to see in lab this week. This is actually fish blastula. So when we do the fish blastula slides, you can see the roundness of the cell. There's one there. Here's one here. And you can see this is the nuclear material. Okay, so those are the nuclei. The DNA inside here is in the form of chromatin. That's chromatin, that's chromatin. And that will have just went through cytokinesis because it's pinching into two new cells. So this is kind of the shape of the lab that we're going to be looking at with the fish blastula. All right, here's an overview slide. These are great, great pictures. You have this in your textbook, so hopefully you're doing your reading and you can see this. But this just summarizes from beginning to end. Okay, so here's G2 of interphase. Now, if you want to know how I know this is G2 and it's not G1 or S, look right here at these double pair of centrosomes, okay, which has centrioles inside of there. So because they've been doubled, I know that this is in G2 and it's not in S and it's not in G1. Because in S and G1, the cell would only have one pair of those. Okay, so the reason for the doubling, if you'll notice in the pictures as it progresses from here to here, one of these centrosomes is going to move to one side of the cell, one of these is going to move to the other side of the cell. So here in Prometa, they're opposite of each other. Here in Meta, they're opposite. Ana, opposite. Telophase and cytokinesis, one goes to each new cell. So after that, then the next phase is it goes back to G1. So each of these cells would be in G1 and have one centrosome. So I know this is G2 because those centrosomes have been doubled, okay? So now let's go into the actual, what I call PP-mat. Okay, PP-mat is prophase, prometaphase, metaphase, anaphase, telophase. Now this phase right here, prometa, is pretty much reserved to AP Bio 2 and reserved to colleges. For freshman biology, you don't really include that as a phase of its own, but they've recently added that because there is a little bit of a difference between general prophase and between metaphase. There's like an in-between phase here, so they've named it prometa. So if you look at the name, it's like in between prophase, in between metaphase. Okay, so during this time, just so you know, the cell is totally worried about division of the nucleus. So all other activity is ceasing during this time, and all energy and all resources are completely used for mitosis. That's what the sole purpose during this is, to make sure that we divide the nucleus evenly so that our cells at the end up with equal amount of DNA. Okay, and then of course, we're going to go through the last step after PP-mat, and we're going to divide the cytoplasm, where we're going to split the cell into two. Okay, so now, if you look at, let me get back to a clean slate here. If you look at this picture that you see right up here, I wanted to spotlight on this for just a minute before I get into all the action, because I'm going to be using words like chromatid, centromere, chromatin, okay, and I might even use the word chromosome. So a lot of C words that refer to the same thing. Okay, this is all referring to DNA plus its associated proteins. Okay, so all those words are referring to this. Okay, except for this one, which is centromere. So in this picture, okay, you see how traditionally we use this little X pattern to illustrate a chromosome? If it's paired like this, that is one chromatid right here, and that's another chromatid, and they are replicated. They are identical. Okay, those are identical chromatids. So the word we put in front of it is sister. Okay, we call it a sister chromatid if it's identical copies. Where do those identical copies come from? Think about what we just talked about. During S, which is synthesis, of interphase, we had equivalent to 46 chromosomes. Okay, but they were all loosely arranged as this chromatin material. So that doubled, like this. So then we had two sets of it. Well, what's going to happen through mitosis is in this set, it will start to thicken and coil and condense, like this, just like you see in this picture over here. Thickens, coils, and condenses, and will form a single chromatid, like that. So that will do that in this one set. Over here, the same thing will happen to that set. And so what you will have is chromatid number 1 pairs up with chromatid number 1 of this side. So this would technically be chromatid number 1 with its replicated chromatid number 1. We would call them sister chromatids, but they represent, guys, this is important, they represent a single chromosome. Okay, they represent a single chromosome. So each of these is really identical, and the destination is one of these will go to one cell, the other one will go to the other cell, but they are identical replicates because they copied over an S of interphase. Okay, but their goal is to go to the opposite cells. So we're still going to refer to them as sister chromatids, but technically they represent a single chromosome in this picture. Okay, so I just wanted to make sure I laid that foundation so that now if you look at the picture down here, okay, look at the overview, you can see in this picture, here's G2 of interphase, this is chromatin, right here, look at it, you can't see any X's, so we call that duplicated chromatin, double set. In prophase, this would be sister chromatid pair there, sister chromatid pair here, sister chromatid, sister chromatid, sister chromatid. Okay, so in us, because this is obviously not our cell because there's not enough of these, in our cell, what it would look like here is we would have 46 pairs of these, like that. Okay, we would have 46 pairs of those, replicated material. In prometa, between prophase and metaphase, these sister chromatids are together, here they are, together, 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 and they are moving around because now they're interacting with these yellow fibers, which are called microtubules. Okay, they're microtubules, which are made up of protein. Okay, in metaphase, look where they are. Sisters are lined up in the middle of the cell. That's why I like to say M for meta, M for middle, because they're in the middle. Okay, look at, and by the way, in us, there would be 46 of those pairs right there lined up in the middle. Okay, in anaphase, sisters pull apart. Look at this. They're pulling, and they're going to move away from each other, and so they're going to take on a new name. Okay, now the name is not called a sister chromatid because as soon as they separate, these over here are destined to go to one cell, these are destined to go to the other cell. So they grow up, and the name that we use is now these are called daughter chromosomes. Okay, but they're individuals. That's a chromosome, that's a chromosome, that's a chromosome, 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 chromosome. So those are individual chromosomes. So in us, there would be 46 of those moving this way, 46 of those moving this way. So in the end, over here, this new cell and this new cell in telophase and cytokinesis would have the equivalent of 46 and 46, but these individual chromosomes will start to uncoil, unravel, and go back to chromatin. They will go back to how they came from so that the cell now enters interphase, and this will be back into chromatin form. Okay, so that's the big overview. So now let's take it slide by slide and go into so that you can break this apart and see what you see. Okay, so if that was too much for you, no big deal, because we're going to look at the whole thing right now together. All right, here's the cell at the end of interphase. Okay, this is called a centrosome. This is called a centrosome. Okay, you might want to write this definition down. Okay, a centrosome. Okay, a centrosome is composed of centrioles plus aster. Okay, and by the way, aster in Latin means star. That's what that means in Latin. So aster is this. Look, I'm going to draw it for you. All these little tiny microtubules around this, those are called aster. It's the beginning formation of spindle. So the centrosome is the centrioles plus the aster, okay, plus also the small microtubules, which are protein. So there's quite a bit of protein that's in there as well. Okay, let me make sure I hit everything on that. Yep, got it. All right, so that's everything that's in that little centrosome. So at the end of interphase, we've already doubled our DNA. When did that happen? That happened in S. Okay, it happened in synthesis. The centrioles are inside of the centrosome region, which is composed of the centrioles, the aster, plus all the proteins that are going to be in that area. Remember, only animal cells have centrioles. Plant cells don't have them. Okay, and then the nuclear envelope and the nucleolus are still here. Do you see how this is still a structure? There's the nuclear envelope. There's the nucleolus. So everything's intact. Okay, so the cell is ready. It's doubled all of its organelles. So inside of here, like all the organelles, like mitochondria, Golgi, ER, lysosomes, if we're talking about an animal cell, Okay, all of the organelles have been doubled. Okay, everything is ready. And so now we're ready to move into mitosis. All right, as far as what the cell would look like, I threw this in here because of our lab this week. These are onion root tip pictures. So when you do your lab this week, this is what you're looking for. You can see the cell wall makes it boxy because the cell wall is made of cellulose. But I don't know if you noticed, but most of these cells look the same. This one, this one, this one, this one. All these cells look the same because they're all in interphase. The only one that I see that's not in interphase is this one right here, which you can see the DNA is kind of thickening. See all these little squigglies, which would be prophase. And then this one over here, to me, looks like it's also in prophase. But the rest of these cells are inter, all in here. These are all interphase because that's what the cell spends most of its time in. So I want to make sure you saw that because that's what we're doing in lab this week. Okay, let's move to prophase. This is the longest phase of mitosis. Okay, not of the cell cycle because the longest phase of the cell cycle is interphase, but it's the longest one of mitosis. Okay, so a lot of stuff going on here. That's why it's the longest phase. And let's just kind of take these bullet by bullet. Okay, first of all, the goal, guys, remember, mitosis, we're dividing the nucleus. So we can't divide this nucleus unless we do number one, which is we have to get rid of that nuclear envelope. Right now, it is a boundary. And we have to get, these are microtubules right here, we have to get those in there to grab onto the DNA because those are responsible for splitting the DNA up to two new cells. So we have to counteract that boundary by dissolving it. We have to get rid of it. So in the background, guess what organelle is back here and will release some hydrolytic enzymes to go up and chew away that nuclear envelope. Hopefully you just said lysosomes. Okay, because that is what is there. So the nucleolus and the nuclear envelope disassemble by the action of lysosomes as well as enzymes, the enzymes that are in there that chew up that nuclear envelope and chew up the nucleolus. Okay, and it has to do that in order for these microtubules to be able to get in there and contact the DNA. Okay, number two, look what's happening. This actually takes a while. The replicated DNA thickens, coils, and condenses, meaning it goes like this. It goes from being chromatin to thickening, coiling like this, into a chromatid, and it finds its matching chromatid, like that. So it thickens, coils, and condenses into sister chromatids that are replicated. Okay, so that technically is a chromosome, because it's visible under the microscope, but it's composed of replicated sister chromatids. Okay, they will join at their area called the centromere. Okay, they will join at their centromere. It's a protein disc, kind of like, think of Velcro, like one of them has one piece of it, the other one has the other piece, and it sticks together to hold them together. Okay, now in addition to that, they will also hold together at various places along the length of the arms of the chromosome by little proteins called cohesions. Okay, they'll hold together by little cohesion proteins. If you think about it, it makes sense. Cohere means stick together. So they're cohesion proteins that hold together those sister chromatids all along the length. So you can see in this picture how there's one, there's the other one cohering, and it's joined at the centromere as well as cohesion proteins through the length of it. All right. Next thing is number three, the centrioles right here, the centrosomes. They actually begin moving away from each other because they want to eventually be across from each other. So they're moving to opposite sides, and as that's happening, we get microtubules that are lengthening. How do you lengthen this? You add protein. Okay, so protein is added to the growing end, added, to make these longer to form what we call spindle fibers. Okay, now spindle fibers are it's kind of the name that we give to the whole length of it, but it's included in this little centrosome unit because the spindle fibers are lengthening from there, and that's where tubulin proteins are accumulated in those centrosomes. Now, some of these spindle fibers we're going to call, look at the name, kinetochore microtubules. Some we're going to call non-kinetochore microtubules. This goes back to another thing I'm going to show you on this picture right here. This is labeled centromere. Okay, that's this union where they're attached together. Each of these chromatids also has a little handle, like this and like this, that are faced opposite of each other. Okay, it's like a little handle. The handle is called the kinetochore. Okay, it's called a kinetochore. They each have their own little kinetochore. Okay, that little handle is going to be really important because when a spindle fiber comes in, the spindle fiber grabs on, wraps around the handle, and attaches. One goes like this, wraps around the handle, and attaches. And these spindle fibers, or like we like to call them here, kinetochore microtubules, are the ones that actually come in and go around the kinetochore of the sister chromatid. Okay, so their job is to come in and go around the kinetochore. So they're called kinetochore microtubules. Their job is to act like a little tug-of-war. They're going to like push and pull and push and pull and eventually move the sister chromatids so that in metaphase they're lined up in the middle because that's an important head count phase where we're going to make sure that all the DNA is where it needs to be. Okay, so that's a kinetochore microtubule. A non-kinetochore microtubule is the ones that are going to not touch the DNA, but they will still be there as structure and support, and they kind of will overlap each other in the spindle fiber network. Okay, so some of these are kinetochore and non-kinetochore, and it's based on whether they attach to the kinetochore on the sister chromatid or not. So those will start to extend and lengthen during prophase. Okay? All right, here's a picture of an allium root tip. Okay, so that's a root tip slide. And what you see with this one, same thing, there's the cell wall. Now remember, they do not have centrosomes, so you don't see them down here because they don't have them. But you can see the DNA is thickening, coiling, condensing into its sister chromatids. Look at the nuclear envelope disintegrating. See these gaps that you see where I'm writing with my pen? Those are gaps in the nuclear envelope. So that started to disintegrate in prophase. Look at the difference between that picture and this one. This one's in interphase. Okay, so in the lab this week, that's what you're looking at. A distinguishable characteristic is the thickening of these sister chromatids during prophase. All right, next one is prometaphase. Okay, so you can see a difference. Prometaphase is a little bit further on than prophase. So one of the things is you've got highly fragmented nuclear envelope. Okay, so the nuclear envelope is highly fragmented. In fact, in this picture, all you see that's left of it are little pieces like this. Okay, that's what's left of the nuclear envelope. Over here, it's these dotted lines like this. So that high fragmentation allows the kinetochore microtubules to now get inside of this nuclear envelope and try to get in there and make an attachment to the kinetochore. So you can see this one right here, this kinetochore microtubule is attaching there. It doesn't have the one attaching yet, but maybe this one, as it gets longer, will come in and grab onto this side. So eventually your sister chromatid should be attached by those kinetochore microtubules. So prometaphase or late prophase has more fragmentation. It has even more highly condensed sister chromatids. Okay, so they're more condensed, more visible. We've got kinetochore microtubules attached, and we have the non-kinetochore microtubules like these that are going to overlap each other to create more structure and support. Y'all, you have to have that overlapping because the cell is compromised due to the disintegration of the nuclear envelope. So we need more structure and support in there because we're going to lose the nuclear envelope. Now, I like this picture because sometimes we forget about these old organelles that are in here, but these organelles are also going to have to be split between the two cells. So some of these microtubules will actually attach onto those and pull them to opposite sides as well. Okay? So that's prometaphase. All right, moving on. Oops, didn't want that. Moving on to metaphase. One of my favorites because it's kind of quick. Okay, M for meta, M for middle. That's what I like to think of. So sister chromatids are lined up in the middle of the cell along an imaginary line. That's an imaginary line called the metaphase plate. Metaphase plate. Some people call it the equatorial plane. Equatorial plane, depends on the book that you're looking at. Okay, so sister chromatids line up on the middle. Now this is a very important checkpoint phase because this is when the cell looks to make sure in us, remember we have 46 chromosomes, that we would have 46 sister chromatid pairs lined up in the middle of the cell. And if something's not right, if something's not, going well, the cell will respond to that checkpoint and abort the cell cycle. Okay, it'll go through a programmed cell death or apoptosis. So this is a very important checkpoint phase. Okay, but you can kind of see also, let's just go down the checks here, the spindle fiber network is fully formed. Okay, so it's completely spanning the cell. Centrosomes are opposite of each other. Sister chromatids, attached by their kinetochore at their centromere, are aligned on the metaphase plate, equatorial plane. Okay, and the cell is now ready to go into the next phase, which is anaphase. Okay, so it's a real simple checkpoint. That one's easy. Okay, in the allium root tip, okay, so this is allium root tip slides that you do in lab this week. This is a classic right here, metaphase. Love it, because look at this. They're not perfect, but you can see, like, these are sister chromatids, like this. You can kind of see them overlapping. These are the arms of the chromosomes. And here's the imaginary metaphase plate right there. Okay, that's the imaginary line. This one, to me, also looks like it might be late prometaphase, early metaphase, and they're kind of aligning on the middle as well. But then you can look at these and go, oh, interphase, interphase, interphase. This might be prophase. Okay, interphase, interphase, interphase. You can kind of look at all these. This one down here may be prophase. Okay, this one, I would say telophase, because do you see this? This one right here has a new nucleus, and there's a slight plate right there that's forming. So you can kind of see in these slides, this is what we're going to see in lab this week. All right, anaphase is also one of my favorites because it is quick. This one is the shortest, okay, the shortest of the cell cycle. It's very, very fast because all that we're doing here, and I say all that we're doing, it's very important, is you can see we're pulling apart the sister chromatid pairs. Okay, so here is one of them. This poor little sister lost her sister, sad face. They're long-lost sisters that won't see each other again. Now we give them a new name, and we call these individual or daughter chromosomes. Okay, individual or daughter chromosomes. And these individual daughter chromosomes will go to one new cell, and these will go to the other new cell. So we call these individual or daughter chromosomes. You can see it's listed right here. And each one has its centromere attached, and that's still where the kinetochore microtubule is pulling it from. And they've actually done some research as to where the shortening happens on this. It was kind of a cool little research study. At first, they thought that the shortening happened down at this end, that these spindle fibers would get shorter from here. They actually discovered that it was right through here that the shortening happened. And so the tubulin proteins are shortened and broken off like this. So it's shortening as this is moving further. And then they're disintegrating as they're in the cell because they're not needed anymore. So these spindle fibers are doing major shortening as it's bringing daughter chromosomes to the poles. Okay, so the centromeres had to split for that to happen. So centromeres split, sister chromatids are pulled apart. We now call them daughter chromosomes. The spindle fibers are shortening, okay, as these are shortening by breaking these down right in here, and then the cell is elongating, preparing for telophase, which is where we're going to form two new nuclei. So you can see this like football-shaped cell as it's preparing to go into telophase. Okay. Let me make sure I hit everything on this one. Yep, kinetochore microtubule shorten, cell elongate. By the end, this side will have equivalent of one set of chromosomes. This will have equivalent of the other set of chromosomes that will go to each new respective cell. All right, here's a cool picture. This one is fish blastula, y'all. Okay, so this is the cell, which you can see, this is not a cell wall. It's got a more roundish shape to it. See that? Here's your chromosomes, daughter chromosomes. Look at these kinetochore microtubules, and there's your centrosome right there. Centrosome, those are your kinetochore microtubules. And you can't see the organelles because we don't have that high-powered of a microscope for this one. I thought that was a cool picture. Okay, zooming on down. We're coming on the home stretch. Hang in there, y'all. Okay, now we've got telophase. T for tele, T for two. Okay, not two new cells yet. It's two new nuclei. Okay, new nucleus here, new nucleus here. So we've got a complete set of chromosomes at each pole of the cell. We've got a complete set of chromosomes here, complete set here. Spindle fibers are disassembling and disintegrating. You can see there's barely any left. All we have left is going back to this aster around the centrosome. The new nuclear envelope is reforming. Okay, now if you want to know, well, how does that reform? Pieces of the old mother nuclear envelope come in, and the cell uses the endomembrane system to build a new nuclear envelope. So that starts to reform around each new nucleus. Chromosomes, look at this, are uncoiling, unraveling, unfolding, going back into chromatin form. So this will go back into chromatin because the cell is, after this, going to go to cytokinesis and back to interphase. Nucleolus also reforms. Ribosomes start to do their protein production. Metabolic activities start to resume. In other words, back to like going through protein synthesis and cell respiration, back to doing normal activities, as the cell is now preparing to split in two and then eventually go back into interphase. Now you can notice this, I want to make sure you know this, cytokinesis can begin as early as late anaphase. Okay, it can start in late anaphase to telophase. So this picture right here already has cytokinesis occurring. That's what this pinching in is right here. As soon as you see that pinching in, that means cytokinesis has already begun to divide the cytoplasm. Okay, so that can begin early, but it's going to finish on its own after telophase has completed. Okay, so let's look. Here's a perfect classic picture. This is root tip. This is the plant. Okay, so notice you don't see any centrosomes because they don't have any. But you can see this is a new nucleus here, and you can see what's left of the microtubules, and you see this line, because in a plant, they form a cell plate, which forms the new cell wall. So this is definitely telophase and cytokinesis in a plant cell. Okay? All right, last but not least, here we go, home stretch. Let's go through the ending part of cytokinesis. Okay, so cytokinesis in animal cells, which once again is division of cytoplasm, we have to divide the organelles and we have to divide the cytosol. So what happens is there's actually a ring. Okay, there's a ring of microfilaments. And it's a contractile. It's a contractile ring of microfilaments that forms right here. Okay, so these are still microfilaments that are in the cell that it forms, which are made up of protein. They kind of work with the protein myosin to like pull on each other. So it forms this ring in here that's almost like a drawstring. That's the best way I can explain it. So if you pull on a drawstring and it squeezes and pinches, that's what's happening here, and it's pulling in, so that this is pulling in, that's pulling in, this is pulling in, that's pulling in like that, and eventually the cell is creating this dip. Okay, the name of that dip is a cleavage furrow. Okay, that's that dipping in right here. Just like, yes, cleavage, you guys know what cleavage is on a girl? That's the dipping in, that's a cleavage furrow in an animal cell. Here's a true picture, I love this picture. You can see the contractile ring pulling in right there, creating a deepening cleavage furrow. That'll eventually go thicker and thicker and thicker, and eventually that will be one cell in one cell. So when it's done, here's your two new cells. These will go, and these will be in G1 of interphase, both of these cells, and they will have identical DNA, like this, identical, same number, same amount, exactly. In us, this would be back to 46 chromosomes in each new cell, with its one centrosome with centrioles, and the cell would be in G1 going back to regular metabolic activity. Okay, that's at that part. Now, a difference between that and a plant cell. Okay, look at the plant cell. Because they have a cell wall made of cellulose, they cannot pinch in. They don't have that contractile ring that pinches in. They instead have to use organelles right in here called the Golgi apparatus. Okay, they use the Golgi, which uses vesicles to produce more cellulose. Okay, so the Golgi produces cellulose, packages it in a vesicle, and watch what it does. I'll draw it for you. It travels up here, lays down a vesicle-filled packet, lays down another one, lays down another one. So eventually, a bunch of vesicles are laid down. Okay, more is produced from this side, because both of them have Golgi, so this comes in. This cell plate gets thick. Okay, so it's called a cell plate because it's not yet a cell wall. Once it gets thick enough, and it forms a thick enough area, and there's enough cellulose in there, that cell plate then becomes the new cell wall material. Okay, but that takes a little time. So you can see in this picture down here, there's your vesicles, 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 cell plate gets longer, eventually extends, and there's enough cell wall to support both cells, for both the new cells. So that's one of the primary differences between those. And then here's a picture. You can see classic cell plate. That's a new nucleus. That's in telophase. New nucleus with the cell plate formation. So this is telophase and cytokinesis happening there. Okay? And then I think the last slide is this one, where you can see it kind of just all put in one picture. Here's, this one is interphase. It's actually kind of interphase / early prophase, I would say, for that one. This one is pro to pro-meta. This is definitely meta. This one is ana. And this is tele and cyto. Okay, so do you see how in this picture I kind of said it was early prophase? Because I see disintegration right in here in the nuclear envelope. So I would lean more to prophase for this picture. This one is pro to pro-meta, hasn't really formed in the middle. This is definitely meta because of that metaphase plate. This one's ana, because you can see those are being pulled that way and this way, and there's a separation in here. And then you've got telophase and cytokinesis. All right, I think that concludes it. Yep, because my next video will have those in there.